Good morning, student. I, Nilakshi Komte, welcome you in our science spirit. Today we are going to learn about cell and cell organelle. Last year we studied that the cell is structural and functional unit of living organism. According to the functions, cells of different sizes and structures are found in different organs. There are two types of cells in living organism: prokaryotic and eukaryotic. So first of all, we are going to study difference between prokaryotic and eukaryotic cell. Prokaryotic cell generally smaller in size than the eukaryotic cell. That is, the size of prokaryotic cell is about 1 to 10 micrometer. The size of eukaryotic cell is about 5 to 100 micrometer. In prokaryotic cell, membrane-bound organelles are absent. But in eukaryotic cell, membrane-bound organelles are present. The chromosome in prokaryotic cell is only one, but in eukaryotic cell, more than one chromosome are present. The nuclear region is not very well defined and is called as the nucleotide. But in eukaryotic cell, the nuclear region is very well defined in form of separate membrane bound organelle called nucleus. This is the difference between the eukaryote and prokaryotic cell. Now we go towards actually the structure of cell. Observe the diagram and also observe the name which are given to the diagram. This figure is about plant cell and animal cell. You know that there is a main difference between the plant cell and animal cell is about cell wall. In animal cell, cell wall is absent. Only have cell surface membrane. But in plant cell wall is present. In animal cell, chloroplast is absent, but in plant cell, it is present for photosynthesis. Vacuoles in plant cell not usually present, and if they are present, they are small and temporary vacuoles sometimes found. But in plant cell, vacuoles are large in size and so less in number, and they are large due to the fluid is filled into that. So due to the vacuoles are present or cell wall is present into plant cell, they have fixed shape. But in animal cell, the shape is changed and sometimes they are rounded. Now we go towards components of the cell. First component of the cell is cell wall. Cell wall is found in plant and bacterial cells. Animal cell lack cell wall. Cell wall is an elastic and strong coat around the cell membrane. It is mainly composed of carbohydrates like cellulose and pectin. It is located outside of the cell membrane and made up of cellulose. Next component of cell is a plasma membrane or cell membrane. Cell membrane is also known as plasma membrane, and it is a semi-permeable membrane that separates the inside of cell from outside environment. It is made up of lipid bilayer, which is double layer of phospholipids and cholesterol, as well as proteins. The cell membrane structure and function are critical for cell. Structure of plasma membrane, and it is a selectively permeable membrane also, allows some substance to enter the cell, while prevent other substances. And due to the selectively permeable membrane which is present into the plasma membrane, the two processes takes place into the cell, that is diffusion and osmosis. Here, according to the figure, diffusion is movement of molecule from higher concentration to lower concentration. Both solute and solvent move. In osmosis, movement of solvent across the semi-permeable membrane from higher level to the lower level of concentration, and it is only solvent moves. For there are different types of the osmosis takes place into the solution. Isotonic, hypotonic and hypertonic. That is a term used to describe the concentration of solutes and water in chemical solution. That is, we correlate this term with each other. So first of all, we see about isotonic solution. In isotonic solution, medium outside and inside the cell has same proportion of water. Water does not go in or out. Second, hypotonic. Cell has less water than outside medium, so water enters cell. For example, if resins are kept in water, after some time they swell and become turgid. Then hypertonic solution. The cell has more water than outside medium, so water comes out of the cell. For example, if Fruit pieces are kept in thick saturated sugar solution. The water from fruit pieces enter the sugar solution so that fruit pieces shrink after a while. In this way, if plant cell or animal cell is kept in hypertonic solution, water comes out of the cell by process of exoosmosis and there is a contraction of cytoplasm takes place. So this process is called plasmolysis. Means here the hypotonic, isotonic and hypertonic solutions are depends, that is a flow of water from higher concentration to lower concentration. Now next, cytoplasm. Cytoplasm is a gel-like fluid that fills the inside of the cell but outside of the nucleus. Means cytoplasm is the fluid between plasma membrane and nucleus. It is moving sticky substance. Many cell organelles, for example Golgi complex, mitochondria, are suspended in cytoplasm. Cytoplasm is the medium for cellular chemical reactions. The part of cytoplasm other than organelles is cytosol. Means where organelles are absent in cytoplasm, this part is called cytosol. Cytosol stores vital substances like amino acids, glucose, vitamins, etc. In animal cell, cytoplasm is more granular and dense, but in plant cell it is thin and mostly pushed to periphery due to the larger central vacuole. When we see structure of cytoplasm, cytoplasm is highly organized and heterogeneous with the framework of protein scaffolds called the cytoskeleton. In cytoplasm, there is presence of water, salt, protein and other organic molecules. When we see the functions of cytoplasm, cytoplasm is the most chemical reaction in the cell and it's where cell growth, expansion and replication take place. Other cell organelles which are present into the cell, we will see that cell organelle in our next lecture. Thank you and good day. Now